Hi, everyone. So I really could not be more excited, first of all, to see you all here, and second of all, to be introducing Dr. Uma Ramakrishnan for one of our plenary talks. Uma received her BS from Bangalore University, followed by a master's from the University of Pune and a PhD at UCSD. She then went on to do postdoctoral work at Stanford University before landing her current position at the National Center for Biological Sciences in Bangalore in India, where she's been since 2005. So Uma has made a name for herself working on genetics and genomics of the unique and diverse vertebrate fauna of India, and in particular on tigers, which she's going to tell us about today. And for me personally, Uma has actually played a really important role in my journey. I first met her almost 10 years ago after I saw her give a talk also on tigers at the Field Museum in Chicago, where I was at the time, which totally inspired me to get into conservation genetics. And Uma then helped me figure out where to do a PhD, which landed me at UCLA and has brought me to where I'm at now. So Uma, thank you for being a great mentor for me and for inspiring me. And I'm really excited to hear about the last 10 years of your tiger research. I think Uma is also a really fitting plenary speaker for this conference in Princeton, because for those of you who don't know, the Princeton mascot is a tiger. And also the timing of this worked out really well because Uma was recently awarded the Molecular Ecology Prize. And they asked us if we could present her with the plaque that comes along with that prize here today. So I'll read a quick blurb that they gave us to go alongside it. So the 2023 Molecular Ecology Prize has been awarded to Dr. Uma Ramakrishnan, Professor of Ecology and Evolution at the National Center for Biological Sciences in Bangalore, India. Dr. Ramakrishnan is best known for her studies on tiger and other large mammals, which are the poster children for India's natural resources. Her studies have included census estimates from combined camera trap and genetic surveys, historical inferences from genetic data and museum specimens, and demonstrations of gene flow impacting genomic variation and inbreeding depression. Dr. Ramakrishnan's research has led to valuable conservation applications, for example, by providing evidence used in a Supreme Court ruling that wildlife corridors must be included in certain highway expansions. Dr. Ramakrishnan's work on science communication and community service is exemplary, and she is an important role model to scientists around the world. So please join me in congratulating Uma on this award, which comes with your award. Right.
So thanks everyone for being here. It's really great to, and thanks for that wonderful introduction and for allowing this award to be given here. So I'm Uma and I work on tigers, and it's really nice to be at this meeting because we are also like the tigers, a bit isolated in terms of doing pop gen in a place where there isn't really a lot of it. Okay, so I'll get started. So, basically, despite dramatic kind of world global interest in conservation since 1972, for example, when CITES first started, what we're faced with today is global declines of populations and high extinction rates, right? And so as biologists, while this is in the media, you read about it in the newspaper all the time, and it's kind of triggering, as biologists, the question is, what can we do about it? How do we understand extinction, and what can we actually do about it? So, people have worked on this for a very long time. Ecologists, conservation science is a crisis discipline, as it was defined as a intersection of ecology and evolution. And for example, there's a lot of studies which show that small populations have high probabilities of extinction, often with some threshold sizes, for example, of 100 individuals. At the same time, oh, sorry, this doesn't work. Yeah. We also know that isolated populations, so population fragmentation and isolation of populations, also results to higher probabilities of extinction. So here you see basically time to extinction is higher when you have a few isolated populations. So basically, together from ecology, right, we know that small and isolated populations have high chances of extinction. But we also know that, very broadly, genetic variation and low genetic variation correlates with extinction. This is one of the classic studies from really long ago with butterflies, which showed that irrespective of demography, right, irrespective of demographic stochasticity and environmental stochasticity, genetic stochasticity did lead to extinction in populations with lower heterozygosity. But that's kind of vague, right, this idea that variation is important. And I think it's only more recently that we've been unpacking this a little bit better. But basically, as a summary, extinction depends on population size, genetic variation, and isolation. So this was kind of very well laid out and shown in this recent paper, Zoonomia Consortium, which showed that actually in populations which are endangered, a proxy for those which are likely to go extinct, right, have lower genetic variation than those which are not threatened. And it also showed that this is potentially because of a demographic decline. So changes in population size, decrease in population size correlates with lower genetic variation, which correlates, with a higher potential for extinction. And basically we know that theoretically in these small populations, drift and inbreeding, drift stochastic effects and inbreeding, basically mating between relatives, exposes deleterious mutations, which causes loss of fitness, and loss of fitness followed by lower lambda or growth rate, followed by then extinction. And there's been a lot of work recently, several of the people who are here, Chris, for example, and others, which show that basically, or unpack this concept of genetic load, that basically you have, once you have a bottleneck, you have basically these deleterious mutations, which either, are by chance increased in frequency through drift. This is called masked load. They're present as heterozygotes, or because of inbreeding, they are unmasked and you have fitness consequences. And basically this is based on the idea that most mutations are slightly deleterious. We know this from other species, the distribution of fitness effects, and the combined effect of all these kind of bad mutations results in what we see. So this also has been kind of unpacked a bit better with some long-term studies on species like killer whales and soy sheep. For example, genomics helps us actually sequence genomes and look at runs of homozygosity. These are good proxies again for inbreeding and demographic decline. The longer the runs of homozygosity, the more recent the inbreeding. Shorter runs correlate with demographic bottlenecks, and basically also these studies actually go further to try and link inbreeding or low variation to fitness effects. So for example, this study shows that lamb survival is lower for individuals with high FROH or high inbreeding coefficient. So FROH is like genomic or runs of homozygosity quantified inbreeding, and that correlates with FPED or F pedigree based or FIS or whatever. And similarly, this really nice study which came out just this year shows that killer whales with high FROH, right? So basically long runs, 10 MB long runs, have lower survival, and basically this is because of inbreeding depression. So what? So that's nice. It's like a class we just did, which you all probably know, but it's just nice to situate ourselves. So what about tigers? How do they fit into this scheme of things? I've basically used tigers as a model system to study processes that might be important for evolution. Basically future evolution, hopefully, and not extinction. So this is the historical distribution of tigers in kind of the lighter shade, and it's pretty amazing because tigers are not a specialist species like the northern spotted owl, which needs really old growth redwood forest or something. They're really generalist species which live from really cold tundra type environments all the way to tropical rainforests, right? But because of various human effects, anthropogenic effects, their distributions are much smaller. They've shrunk, and today tigers occupy about 7 to 5% of the historical range that they did. Also, we have already seen extinctions in the 1980s, the extinction of the South China tiger, the Caspian tiger, the island Southeast Asian tigers, 2 subspecies which also went extinct. So basically what I'm going to do today is kind of summarize a lot of the work we've been doing for many years, but I'm going to try to hark back to those concepts of small population size, isolation, and genetic effects which may drive extinction. We're going to ask, or I'm going to show you what the populations actually are, what actually makes for connected populations or connectivity on the ground, spatially explicit, and what impedes connectivity, what could impede connectivity in the future. So in terms of the genetics, what happens to these small and isolated populations? I'll show you 2 examples, one of drift and the other of inbreeding, and then basically something we're hoping to work on in the future to really bring this home to a much more, not just correlations, but maybe we can hopefully get towards saying more than that. So just a little bit about how we work. How do we study tigers? So I wish we could catch tigers and get blood from them. I have a lot of tissue envy, I was telling someone today, but we can't. We have to mostly work with non-invasive samples. India is extremely prohibitive in terms of permissions, and so we work with what we get. So it's mostly fecal samples, and you'd be surprised when you walk through a dry deciduous forest in India, the thing you encounter the most is tiger scat, on the road. So it's not so bad, especially in high density populations. But more recently, we've also been working with hair, and I'll talk a little bit more about that. We do work with some blood samples, but not that much. So first, to situate Indian tigers in the context of tigers globally. So basically, this is some work we did a few years ago where we managed to get data from other tiger subspecies, and there's also similar work by Shujin's group a little earlier than this, which shows similar results. But basically, when you look at tigers across their range, and this is several genomes at different depth, what you see is that you have a strong population structure which corresponds with geography. So these are the Amur tigers or the Russian Far East tigers. This is basically Indian tigers, Malayan tigers, and Sumatran tigers. Our study was missing those from Thailand, and those data have also been incorporated into more recent analyses, but the results remain the same. Basically, you find that these are all distinct populations. Just notice these guys over here, they're slightly admixed, and these are samples from over here. So basically, it was obviously a continuum of variation. The northeast of India pops up in many other species as well as being sort of admixed with Southeast Asia. This plot also shows you the mean number of alleles, private alleles, and you can see that as a function of sample size, because we have very different sample sizes for all these, and you can see that Indian tigers have way more genetic variation. In fact, we're not even saturating with the samples we have. Here I show you runs of homozygosity, and basically you can see that while Indian tigers have the highest amount of total variation, right? There are several Indian tigers which also have long runs of homozygosity. Okay, so basically what's interesting then is 75% of the world's tigers, the numbers, actually do live in India, and also they seem to have most of the genetic variation of the species, but tigers in India also appear to be homozygous or potentially inbred. So why is that? So India is clearly very large, has lots of habitat for tigers, but in the last 300 years, this is 1700 and this is 2000, you can see that, so if you assume tigers are associated with this green, you can see that the patches where they may occur is really fragmented, right? So potentially this process of anthropogenic change, land use change, has resulted in tigers basically on islands of habitat. So while the total number of tigers was large, resulting in high overall genetic variation, today tigers actually are in smaller pockets. And just to let you know that we keep having these tiger censuses based on camera trap data, so they can be identified based on their stripes, and we can use mark recapture methods to estimate a population abundance. And there's no tiger population which has more than 300 individuals, and more, like I think there's 5 with more than 150 or something. So all of these populations are small. The median size is something like 35 or 40 maybe. So what are the populations? That is like whatever, the median size is the number of tigers in a protected area, right? But these protected areas are in clusters spatially. So we recently tried to synthesize work from across many years. We started with microsatellite data in 2009, and now we have whole genomes. So basically the picture is not very clear, but I'll show you. These are just structure plots based on the genomes. What you see, for example, here is that these guys are a population, these guys are a distinct unit, and all of these guys are a distinct unit. But as K increases, you see more spatial separation. And when we have a higher sample size but much fewer markers, so these are fecal samples and SNPs, you see kind of similar patterns but much more detail, because the sample size is often much higher with non-invasive samples. But basically, just in summary, because I don't want to bore you with the details. Overall, we have 3 to 6 populations, and these are landscapes, right? So they're not individual, they're not always individual protected areas. They reflect geographic landscapes. That makes sense, that's good. But we seem to have larger and connected populations in the central parts and southern parts of India, which is the Western Ghats, the one place where development is hard because you have mountains, okay? And there are already some populations which appear to be isolated, and I'll talk more about both of these, Ranthambore and Simlipal. And there are, of course, still certain population relationships and connectivity which seems we still don't understand, because we just don't have enough sample size. For example, between the foothills of the Himalayas and the Northeast, we don't really understand that connectivity yet so well. Okay, so basically I'll just show a little bit of work about connectivity first. So we did some work, my student Prachi did some really nice work in central India. So this is, I'm just situating you here in the central part of India, and these are forest fragments, and these are the black things are the protected areas, which are tiny. So just to give you context, 5% of land area in India is protected. All of India's protected areas together are 3 times Kruger National Park. So they are tiny. These are really, really small areas. These are big animals that we're talking about, right? So basically, this is just to cut to a chase. Basically, landscape genetic approaches allow you to correlate land use types with genetic differentiation in some sense, and infer resistance to movement provided by land use elements. So for example, here we have agriculture, semi-urban areas, urban areas, forest, and so on. And this is what the resistance map looks like. Basically, the darker colors makes it harder for tigers to move, and the lighter colors makes it easier. And you can see, for example, that we have the road network really well highlighted, and cities in dark as well. So it's actually good news, because what this analysis suggested are that in general populations are connected. Tigers are moving, But there definitely are barriers, and those are high traffic roads and high density human settlements. And this is good because this is what we find for all species, in a sense, globally, right? So also, if you just look at how both isolation and population size affect extinction, these are just those protected areas we saw earlier, and the colors are basically extinction probability for a given scenario in the future. And what you see is, as you would expect, small and isolated populations always have high probabilities of extinction. Large and connected populations, large is, they're not really large, but larger, have lower probabilities of extinction. But there's a whole lot of stuff which is in between, right? And these are where one could think of management intervention. And what should you do? Should you try to increase connectivity, push them down on this axis, or should you increase population size? Which kind of intervention might actually minimize extinction? This is the hope we had while doing these analysis to help managers to think about these kinds of questions better. But overall, basically, also important is this is a metapopulation, right? So the continued survival of many of these small populations is sustained only by gene flow. So we will see extinctions and we will see recolonization, but increase in barriers in the future will definitely impede connectivity, and this is something which will happen with development. So as Chris mentioned, we were really lucky that at the time we were doing this work, these are two protected areas in central India, Kanha on this side and Pench Tiger Reserve, where Rudyard Kipling wrote Jungle Book, is on this side, and this highway, which was 2-lane, was being expanded to a 6-lane highway, and that is definitely going to be a barrier. So we actually modeled what would happen to gene flow and extinction given this became a barrier, and showed that extinction probability would increase substantially and so on. And luckily for us, we didn't do more than that, but citizens took these studies, these papers, as a public interest litigation to court, and they were able to get basically mitigation structures kind of granted by the court. So these are probably the largest mitigation structures for connectivity in India, and you can see these are these are not high tech, right? They're not like funneling or something like that you have in Banff. There are people everywhere here, but what's really cool is recent studies, camera trap studies, show that tigers are moving across, prey are moving across. So whatever it is, it works, and connectivity is hopefully going to be maintained between these two protected areas. So if we want connectivity, we basically want tigers moving through landscapes filled with people, okay? India has a lot of people, but it has a lot of wildlife, so a lot of tolerance for wildlife. And what that means then is that tigers are going to interact with people. Tigers are huge, okay? Imagine having a tiger in your backyard, and this is what these people are actually living. It's a really fun series of pictures. These are camera trap pictures. This is a female. You can see her at 9:15 in this place. It's outside PA, outside protected area, and another one here with her cubs, okay? 11:30, and here she is in the morning at 7:40 with her 3 cubs, and the same spot at 12:45. People walking, doing their whatever, like collecting firewood and so on. So these people are really overlapping with tigers, okay? And this leads to what we call conflict. I'm not going to call it conflict. I'm going to call it negative interactions between humans and tigers. And what that means is basically livestock depredation. In some cases, there are attacks on people, and that does happen also a lot. And usually in those cases, the tigers are immediately removed. But livestock depredation happens on a daily basis, okay? So one of the parks where I'm going to show you the results from we worked, basically there's up to 4 cases of livestock depredation a day. And this particular state spent, I think last year, $1.5 million on compensation to people who lost their livestock because of tigers. So basically we decided, oh yeah, so if you wanted to study livestock depredation, how would you do it, right? So I'm just going to show you this nice video. I'm sure if you have a cat, you probably know these are two cubs, and that's a kill over there, okay? And this one is like watching the camera, you can see. So they're really curious. And you can see that the cub is really handling the prey, batting it around, licking it, biting it, and so on. And because of this handling of prey, so this is just another picture to show you that what happens is you basically have bite marks. Can you see the bite marks in the jugular? And around these bite marks, you actually have a lot of hair as well. So basically, this is actually new work which is not yet out, but we're really excited about. If you look at a kill site, you can treat it like a investigation in some sense. You have bite marks, you have lick marks, you have hair which has been scratched off when the animals eat, right? And you also sometimes have fecal samples around the space. So what you could do is, what we did is to sample kill sites. We sampled 145 kill sites around a park and asked simple questions. Are there problem animals? So that's the dominant narrative. There's this one tiger, bad tiger, it's going around killing all these wildlife. And I think some of these narratives stem from like Jim Corbett books and stuff like that, right? So are there problem animals? What are the patterns? And maybe can we do anything about it? So basically, we sample these things, bring them back to the lab, extract DNA, ID species, genotype individuals, and then look at patterns, right? So this is the park we worked in. It's in Madhya Pradesh. It's called MP, and I'll just orient you. But these are basically all the kill sites, and the circles are how many individuals were found at that kill site. And basically what you can see, I mean, this is just a graphical representation, but I think this is more telling that most individuals were found at 1 or 2 kill sites. Okay, and that is totaling to something like 40 individuals. This park has about maybe 80 or 100 individuals. So 40% of the animals in the park are actually killing livestock. There are a few individuals, like this one individual, which is at 7 kill sites, potentially a problem individual, right? But this is just an ecological response. There's a lot of cattle outside, animals, there's a high density of animals, there has been recovery, and animals are just killing cattle, as they would any other prey. We also do see some spatial pinch points, areas where there's more killing and livestock depredation, and maybe these are something we can address. Maybe we don't know why that is. Maybe it's the distribution of cattle or prey or whatever. But overall, basically, this kind of throws some concern on the future of connectivity. So how are we going to maximize connectivity while minimizing human-wildlife negative interactions? I don't know the answer, but I think it's important for us to try and understand what the patterns are. Okay, so now I'm going to talk about isolated populations, and this is work we've already published, and it's work by Vinay, who's here presenting some other work. But basically, for a long time, there's been a mystery about these black tigers. They're pseudomelanic, they are pattern disruption, apparently pattern disruption phenotype, and they were found or camera trapped only in one population in eastern India. So immediately it seems like a potential place where we have genetic drift and isolated population. So the first thing we did was, of course, to look at the literature, and there's been some really nice work by Greg Barsh, Chris, and others looking at patterning in cats. And you can see, for example, that there's a mutation which distinguishes this cat from this cat and this cheetah from this cheetah, and it's basically pattern disruptions, the same thing. The stripes are not laid regularly or the spots are not laid regularly, but you have like a mixing up of the black. So there's one gene which they identified, and so we decided to use that as a candidate, and luckily for us, there was basically a litter born in a zoo. So here you have the mother and all of her cubs. This individual is normal, there's a pseudomelanic individual, and there's a white pseudomelanic. So independent traits. White is also a mutation which is known, the white tiger mutation. It's a lack of orange pigment. So this is not a color variant, it is a pattern variant, right? So we were really lucky to have this litter be born, and then basically Vinay was able to use this pedigree and an association analysis to look at sequences of this gene, and we found a point mutation, a missense mutation, which converted histidine to tyrosine, and basically individuals with this mutation were 200 times more likely to be pseudomelanic. So we assume, or we infer that this phenotype is caused by this missense mutation. But that's in the zoo. Is it there in the wild? So when I went back to the field, walked hundreds of kilometers, collected tiger scat, brought the scat back, extracted DNA, identified species, tigers, identified individuals with a SNP panel, and then genotyped the mutation, and basically it's a recessive mutation, and what he found was the frequency of the mutation was almost 0.6. That's pretty high, right? In this one population, the frequency of the mutation was high, and I'll point this to you. This is not a population estimate, it's a minimum number of individuals, because we didn't collect them in an estimation framework, but that is 12. So this is a really small population. So maybe we missed it. Maybe it's there in heterozygous state elsewhere. We looked at 400 individuals across India and some other parts of the range, and wherever we looked, we didn't find the mutation anywhere else. Okay, so it seems to be absent elsewhere. So why is this? Why does Simlipal potentially have such a high frequency? Well, simple like population structure analysis shows that from other parks in central India, Simlipal is isolated. Individuals in Simlipal are also more related to each other than, say, other populations. And also, for example, this one is the northwest population where we know tigers are isolated. I'll talk about it a little later. But basically, Simlipal is isolated and small. Okay, so maybe it's just genetic drift, right? Why is it so isolated, right? So we can use our understanding of connectivity, which we did before, to model connectivity in this landscape. And basically, depending on this dispersal threshold, if you imagine dispersal threshold is 200 kilometers, there's absolutely no connectivity to Simlipal. If you allow for higher dispersal thresholds of 500 kilometers, only at 1,000 kilometers does it become connected. So that's assuming tigers will disperse 1,000 kilometers. That's pretty far. They do disperse 500, 600 kilometers. It's known, but those are kind of rare cases, right? So basically, it's just too far. So imagine, when you think about isolation, tigers are large. You don't expect that, there would be a lot of isolation. But even for tigers, that metapopulation concept where you have sources and close by sources where individuals can disperse from is really, seems to be really important. Okay, so basically we also just did some very simple simulations to ask how can the frequency of the allele go from near 0 to 60%, and basically whatever scenario you look at, it has to be a very strong founding event where one of the individuals did have the mutation. And we're continuing to work on this population. So I feel like the allele will fix in some time because it's a really small population. And so we'd like to continue to monitor allele frequency every generation, which is about 5 years. And we just did some sampling in January, and we have 120 scats. So we can see whether the allele frequency has actually increased. We're also hoping to look at whether we were not able, so the results are consistent with drift, but we were not able to explicitly look at the possibility of selection, and hopefully we'll get some whole genomes and we can look at that better. Also, it's just it's a phenotype, right? It's something we can see, but does it really matter? So at least from the cat work, there's no suggestions that this mutation, though missense, impacts fitness, but that's something which maybe if we can continue to study this population, we can think about. But what's really interesting also is that there's a, oh, sorry, I didn't realize there was music. Sorry. Okay, I didn't realize there was music. But this is a video, and you can see this tiger is really black, right? You can see this is a really dark individual. So the zoo animals I showed you, they were not this dark, right? So clearly there's also variation in the phenotype, and what we're trying to do, again, pretty crazy, I don't know whether we'll be successful, is to, sorry, no music, is to basically, you saw how that animal was scratching on the tree, so we get a lot of shed hair, and this is Vinay actually collecting these shed hair from this tree. You can see it's like some voodoo magic. You can barely see the hair, but it's there. And actually, in earlier work, Anubhav, who's also here, has shown that actually shed hair is a better source of genomic DNA than fecal samples, well, to reconstruct genomes, because fecal samples have a lot of other DNA as well. So basically, if we have camera traps, right, and we have a tree in the middle, and we get some shed hair, we can try to phenotype, we can try to get genomes from these phenotyped individuals. Okay, so I'll talk a little bit now about this other population we studied and continue to study, Ranthambore. It's like a quintessential tiger India. So this is basically, this is a hunting lodge. This is Rajasthan. It's close to the desert. It's very dry, arid, and these were hunting lodges which are now abandoned, and you often see tigers resting there in the summer. Yeah, so this is a very famous tigress, walking kind of along a reservoir there. So basically, this population through, and this is just to point out, this work was started by Anubhav, who is here, and Aditi, who couldn't be here because she couldn't get her visa in time, is continuing this work. So basically, all the studies from many years ago on population structure show it to be an isolated population, and we tried to understand inbreeding from a genomic perspective. Here, we compared Ranthambore or Rajasthan tigers, these guys, to tigers from here. This is you can see a larger connected population very broadly, and these guys too are a larger connected population in South India and Central India. And basically, runs of homozygosity on an average are twice as high for tigers in Ranthambore as in these other larger connected populations. What's also important is a lot of these runs of homozygosity are shared. So the idea of inbreeding depression is that deleterious mutations are in these runs. They cannot be broken by recombination, and you have a phenotypic effect because of these shared runs. And so we also see, and this is because of founding. So while tigers have recovered, most populations were founded by very few individuals. And what mutations those individuals have is potentially going to affect the trajectory of inbreeding depression in these populations. I should say that we don't see inbreeding depression yet. We don't see inbred phenotypes, but we do see genomic evidence of inbreeding. We also then looked at load, and basically what we see is the number of damaging mutations correlates with FROH. The higher the FROH, the higher the number of homozygous damaging mutations. And this is also true for loss of function, potential loss of function mutations, but the relationship is less strong because there's less of them. So we also find evidence potentially for purging. This is statistical evidence, right? What we find is that in the small and isolated populations, in the zero frequency bin or the low frequency bin, you have more potentially damaging mutations than neutral mutations. So that means that there's an enrichment of very low frequency for these potentially damaging mutations. That's suggestive of purging, that selection following a bottleneck has potentially removed these mutations. You see this overall across India as well. The signatures are actually stronger pan-India because the sample size is higher. So it's interesting because you have purging. So generally the narrative is if you have purging, you're safe, you don't have to worry about inbreeding depression. But we see signatures of purging, but we also see high frequencies of the remaining damaging mutations which are shared between individuals, suggestive of the potential for inbreeding depression in the future. So what we're doing now is, because I mean without a phenotype, without fitness, at least I feel I don't feel satisfied with these analyses. And so what Aditi started by Anubhav and what Aditi is continuing to do is to try to reconstruct pedigrees in this population. Now, it's perfect because it's isolated, right? You don't have a situation with a lot of animals moving in and out. There's about 70 or 80 tigers in Ranthambore. So over the past, so here again, this is a high density population. We follow animals, we watch them sit or rub. Once they move away, we collect shed hair, we extract DNA and sequence genomes. And basically after about 6 years of sampling, we have 20 mate pairs and 62 individuals. I know it still seems like a really small number, but that is, these are the potential pedigrees. These are the matrilines. There are 9 matrilines in Ranthambore. This is the largest one, founded by a very famous female called Machli. She has a Wikipedia page. She's one of the most photographed animals, tigers. And basically, though, many other matrilines seem to have kind of died out. So what we're doing right now is just initially reconstructing this pedigree. So we basically look at mothers with cubs through camera traps, look at mitochondrial lineages to confirm maternity, look at which males were camera trapped in the area like 2 years before or something like that, their potential fathers, and then we will reconstruct maternity, paternity, and then kind of piece together this pedigree. And then hopefully, if we continue this for another many years, we can ask questions about these mutations specifically, something like segregation distortion. Do we see fitness consequences of mutations? Okay, so it still really bothers me that we don't really know what these mutations do. We talk about damaging mutations with a broad brush, and some of the work Vinay is going to present is what we hope to do in the future, whether we can use all of our biomedical knowledge gained from mice and humans with clinical phenotypes, some phenotype associated with a mutation, to actually kind of look at the mutations we have in tigers. And he's looking at this in the context of a zoo pedigree, which includes, actually, I found a lot of inbred individuals. So I was always looking in the wild, but zoos are really great because they have a lot of inbred individuals in zoos, right? And so our ability to kind of densely sample pedigrees is much better in zoos. We can actually try to look for fitness effects as well. What we'd like to do in the future also is, once we have mutations which we think have consequence, we'd like to validate them in cell lines using CRISPR or something like that to make those mutations and validate them. So this sounds like really crazy molecular biology, and why are we going down this route? I think it has important consequences for conservation, because when I'm talking about introducing individuals or when should we do genetic rescue, if I have a list of mutations which I know are like red alerts, then maybe it can be a little bit more targeted versus a sort of hand-waving argument of genetic variation is good, bring some individual from somewhere, and so on. Okay, and so that's kind of, oh, sorry, I missed one slide, I think. Sorry. Yeah, so we do a lot of work on other species as well, and for about as long as I can remember, I've wanted to work on Asian lions because they are the quintessential bottlenecked population. But Asian lions, they went from 20 or so individuals, which was saved by this erstwhile king of the area, to now 1,000 animals. They're wandering around the whole state. So what's happening with inbreeding and inbreeding depression in this species? Have they just completely purged all high effect mutations, and are we going to see effects of masked load like sometime in the future? I think it's really a mystery to me, and I'd really like to understand that better. We've just sequenced some genomes which suggest that Asian lions are extremely homozygous, and I'd really like to understand this better. And here especially, I think the zoo systems will be very useful because they're so highly inbred. They're almost like an inbred line, and so we could hopefully look at phenotypic effects of mutations easier. We've also just done some work on elephants, which we are hopefully submitting soon. And Asian elephants also show signatures of isolation. Crazy to think about it, right? They're huge. They move huge distances, but we see kind of serial founding and dilution from the north to the south of India, and as you go more south, populations seem to have lower variation. So there's a lot more we're hoping to explore, both with tigers in terms of a deeper understanding and also with other species. So I just should thank all of this work is very, very expensive, and I'm really lucky to have support from various organizations and collaborators and people who without hesitation share samples, and of course the forest department and so on. It's really great that some of our work can actually feed back into outreach. So this is the enclosure of the black tiger and a poster which the people at the zoo have made about recessive mutations and why this tiger looks this way. And I think it's a nice opportunity for us to connect back to explain some of India's biodiversity and the phenotypes we see. Thank you so much to the organizers and to SMBE. It's really great to share our work, and I'm happy to take questions.
Questions for Uma? We have a hand here, and we have a microphone.
Hi, thank you so much for your talk. This is so impressive.
Thank you.
Very cool. So you showed a plot a few slides before that showed evidence of purging because the lowest allele frequency bins have the highest loss of function.
Yeah.
So I might you might expect this in a typical population because the most deleterious variation tends to fall in some ultra rare bin, basically.
Yeah.
So I guess if you were to cut off the most deleterious alleles, I assume you would see the same pattern for slightly deleterious.
Yeah, so the basically damaging is both missense and loss of function.
I see.
Yeah,
Okay.
together. But I take your point, and I think one of the things, again, we're hoping to do over the next few years is to look at historical material, because if there are really low frequency mutations, we should catch them. Also, a collaborator, some work which I started several years ago, and other with collaborators at Stanford have just put up a paper on bioarchive looking at 156 genomes from captive tigers in the US, Ellie Armstrong, Dmitry Petrov, and others. So basically, when we have more and more genomes, I hope that we can really address these frequency issues better. Definitely, it's a problem that we have low sample size. But yeah, thank you.
Thank you.
Thank you. Very exciting talk. I very much enjoyed listening. I wonder about, in the end, it's this wish. So we have the genomic data. We can say something about mutational load, but then obviously the link to phenotype would be so exciting to do. And I wonder from the feces that you collect, can you, for example, say something about the health state of these animals that you could, for example, connect?
Yeah.
Info?
So I think my student Aditi is also looking at chemical cues, and there are some known chemicals which correlate with health status, like whatever, with extreme conditions like cancer or something like that. So that's one option for wild. There's also with the zoo samples, we have the pedigree, right? So we know when individuals died, how old they were, what condition they had, and maybe we can try to look at those kind of effects better. But yeah, I think looking at fitness in the wild is going to be really, really tough. Also because they basically medicate these animals, they catch them, they vaccinate them. There's a lot of intervention. They're not wild, right? So definitely that would be really nice if we could think of. So one of the things I'm trying to pursue is cardiomyopathies, because cats are known to have cardiomyopathies, and we find some missense mutations in tigers which are known to be associated with cardiomyopathy, in genes associated with cardiomyopathy in cats. So I don't know whether there's a way to look at, like, say, postmortems and enlarged hearts and things like that, but it's going to be quite difficult.
A question?
Yeah.
I really loved your talk. It was
Thank you.
incredible. Going back to the plot where you demonstrated purging or evidence of purging, there were damaging mutations that were fixed in 3 of the 4 populations, but the small isolated one, maybe I'm misunderstanding this, was not that much higher than neutral mutations. Is that something we'd expect, or?
Yeah, so basically when we look at loss of function versus missense, we find fewer loss of function mutations in the small isolated population than the others, but damaging are comparable or slightly higher. So basically, if those are loss of function, we do seem to see a higher kind of quote-unquote purging effect in the small isolated population, but you also see it for pan-India overall. So basically that kind of corroborates the range-wide study where we do see high variation, but we do see potentially inbred individuals in India.
First of all, congratulations on your award. It's clearly very well deserved, and thank you for sharing your work with us. I was actually curious about your work with the tiger-human interactions.
Yeah.
And you saw a few individuals with high kills.
Yeah.
I was just wondering, do you have any information on like the age of those individuals and maybe how age corresponds to their hunting?
Yeah, so that's something we need to do a lot more work on that. I think that it could be, so for example, that female with the cubs, right? So she's living pretty much outside the protected area. She is going to eat cattle, and she's going to teach her cubs to eat cattle, to hunt and eat cattle. So there might be some kind of behavioral associations and propagation of this trait, right? But for that we need to do long-term studies, and we need to look at who these individuals, not just how many individuals, but who these individuals are, how they're related to each other, how does that relate to whom we camera trap, right? So also this is all moot unless we can connect a genotype to an individual. So I'm just going and sampling hair or saliva. I can genotype a tiger, but I don't know who that tiger is. So for that I need a database where I collect samples, like say hair samples from phenotype tigers, like whatever, camera trap tigers or something, and then I compare these to that database. Yeah.
On that note, the pedigree data that you're working on looks very exciting. Can you say what kind of data that is that you're using? Is that
genome-wide? So basically, I think out of those 62 individuals, maybe 40 or so, 45 or so are whole genomes from blood or tissue. Animals they caught incidentally, which they shared the samples with us. The others are hair samples, which we are doing whole genomes. Our depth and coverage may be lower.
Are you using a target approach for that?
No, we're not. Right now, we're not doing targeting, but the goal is that if we can figure out some mutations or specific sets of alleles we're interested in, we can then design capture approaches to look at them more. Yeah. See you. Yeah.
Okay, great talk. Yeah, that was wonderful. My question is, and again, also going to someone who asked about like the areas of the tigers that are eating cattle versus maybe the ones that are feeding off more wildlife. And you have so much data, which is great, and especially scat poop. I was wondering, have you done any sort of paired microbiome comparing the tigers that are more in the cattle, high kill areas versus the more isolated ones?
No, we haven't. Okay. And actually, Abhinav is doing some work now on metabarcoding to just look at diet.
Okay.
Cool. And I think I think that we need to do a very proper study. I feel like the proportion of cattle in diet will probably have something to do with park shape. So parks which are very linear or have a lot of edge may have a lot, because there's people everywhere around, right? So they may have more, livestock depredation. It also has to do with policy. So basically in northern India, there is a ban on cow slaughter because of various reasons. And so there's a lot of cattle. Whereas in southern India, there's more cultural consumption of cattle, and some other work we've done on vultures, which are scavengers, shows that vultures have a lot more cow. In pretty much all their diet is cow in northern India, but in southern India, they're more dependent on wild prey. So there may be other things like that, which I would be very scared to publish, but we may get into trouble, but yeah. And sorry, for the microbiome, we've not done it. So with microbiome, I feel sometimes very correlative, and I'm not sure what to expect. So I think if there was a candidate or a set of things we could look for. So for example, with some other microbiome work, we found that commensal species have like lactobacilli and stuff, which the forest or the wild species don't have. So there may be some human associated microbiome which may show up, but it would be interesting to think of hypotheses like. It would be interesting to look at the geographic locations. Yeah.
Great talk. I have not a genomics question, but with the livestock predation, how tolerant are the farmers of tigers? And then as a follow-up, are there sort of lessons that we could learn that might apply in North America where we're not as tolerant of large predators and livestock?
So the farmers are very tolerant, right? So basically, I don't know. I think people in India are generally very, very tolerant. And, one thing which, at least here, is if the compensation system can be speeded up, that would definitely be better. But I think that compensation in general is not a long-term solution. I don't know what the solution is. I mean, a very radical, let's say, sensitive solution would be to not have, cattle outside a protected area, for example, have other livelihoods or something. But, those are very difficult to. The thing is that I think here you have probably large ranches which are protected. In India, you have free-ranging cattle. So people just graze their cattle wherever. And so,
in Western Canada, they'll graze cattle in public land, and it's the issue you're talking about. Often farmers aren't compensated quick enough.
Yeah. So one of the things, for example, there's different compensation for leopard and tiger and so on. So these genetic approaches, if done very quickly, they could potentially, aid in that.
Hello.
Yeah, hi.
I kept thinking about what you said, that in some areas they have been vaccinated, like the tigers.
Yeah.
And I was thinking about this concern that vaccination can actually increase load. Do you think these populations can be used to address that question?
Could be what, sorry?
Could be used to address the question of whether population that has been treated have more load than population that has not.
Basically, I have this collaborator of mine who works on lions. He's an ecologist, Jhala, and he says that he feels that with the Gir lions, so tigers is not so much of vaccination. They treat them if they're unwell or something like that. But with lions, there's been vaccination and stuff, and he thinks that this could definitely lead to more load. But the key would be to look at, so the intervention is definitely a lot more in the last 10 years. So if we could look at samples before, there are some samples around from 1980s. So if we could look at those and look at modern and look at historical, it would be really nice to bin time into different types of processes which are happening. Thank you.
Any final questions? Oh.
It's bad when your student wants to ask you a question.
Just a continuation from the previous question, as I was wondering, even if you find higher load in the lions, for example, would that is it possible to compensate that with the lifestyle of the lions, where they live in a community, right? They live in herds. So groups, maybe the nurturing sort of compensates for
some kind of inclusive fitness.
Maybe because you can have high homozygosities for certain deleterious alleles in individuals, but individuals may vary in what deleterious allele is homozygous in them.
But it's likely that individuals in a pride will have, they'll have correlated ones. But I think that, so I think ultimately the thing about load is, if we want to, if we agree that minimizing load is a good thing, then at least we detail these mutations. We can think of, if we are to do genetic rescue, where from, how much, and so on. So what I would love to do in the next few years also is to try to outline guidelines or some approaches to how to do rescue, and maybe maybe it does depend on life history strategy and so on. But like with elephants, it's very different. Their life history is very different. Their behavior is very different. I think we need a lot more work to understand that better.
I just got it.
Uma, one follow-up question on that. So on the genetic rescue side, is this something that the Indian government is open to and would be willing to do if there's some sort of plan that you propose?
So have you not heard about the cheetahs? Like bringing them like from Africa and all this drama. But I think basically the government of India today is very interventionist. And just recently, Rajasthan, we've been working there on this isolated population for like 8 years now, publishing these papers. And finally, the PCCF, the head of the forests over there, has asked us to comment on, if we are to do genetic rescue, where should we bring individuals from? And so that's why I'm saying, I mean, of course, I have to try and jump at that opportunity and do some good research and make it digestible. Like, okay, if you do this, you have to bring them for many years, or you can't just bring 5 animals and dump them. But the thing which I'm a bit worried about is that I actually don't know anything. I mean, I can't comment on behavior, right? And with cats, I think behavioral integration and stuff is a very big thing as well. So I think that's something which maybe, I don't know, we can think about, like how do we, because if as a geneticist I can say something and I can make a disclaimer, this is only genetics, but what actually happens, I don't know, publishing a paper is one thing, but saying move this animal, that's a very different thing. And I'm a little nervous also about how to go ahead with that. Thanks.
Wonderful talk. I was wondering, are any of the more inbred tigers, do you see those sort of classic cat phenotypes of inbreeding, like the kinked tails or the spinal malformations or recessed testicles, anything like that? Or do they sort of look yet. Okay?
They seem to be okay. So so I think it's something which we'd really like to look at. And one of the ideas actually Anubhav has had is to try to use ML to look at, videos and piece together potentially like a normal morphology tiger and one which is because there's a lot of tourism here, there's a lot of people taking pictures and videos, so we could try to maybe bring some of those approaches. But yeah.
Hi, thank you. To understand more of the phenotypic associations with small populations, have you ever considered like baiting some of the trees in front of the camera traps to get maybe even RNA data to do transcriptomics and understand functional consequences?
Of course, we fantasize about those things, but I don't know whether we would. We can try. So basically, the thing is that in all these places, we have to build long-term relationships, with the managers, and they have to trust us. The good thing about Simlipal, where the black tigers are, is there's no tourism yet. So it's a little bit, you have a little bit more flexibility of what you can do. Like in the other park, Ranthambore, I mean, we can't even get down while the tourists are there because, there'll be a huge hue and cry. So it's a possibility. And that would be really nice if we could. I think that's probably a very complicated question. I don't know whether we'll be able to figure it out, but it would be really nice to try. Thanks.
Okay, maybe one last question.
Yeah, this is an awesome talk. We'll talk a lot later on.
Yeah.
I'm just commenting on the baiting idea because with jaguars and other wildcats in South America, we have similar discussions, and the ecology people actually have issues with the baiting because it disrupts the study design for camera trapping that assumes no baiting. So we want to bait for some reasons, they don't want to bait. So it's like the behavioral issues that Uma already brought up. I mean, there are many other layers, and we geneticists have to take the other aspects into account as well. But yeah, it would be great for some studies.
All right. Well, thank you everybody for the amazing questions. Thanks, Uma.